discussion, presentation. Let me introduce myself. You all know my name is Rakesh Singh, and that's my email address. If you ever have to contact me, if you have any question which you cannot ask in person, feel free to send me an email. I will be happy to respond. Like many of you, I did my PhD here, actually master's and PhD both together with Dr. Kalpana Pai, under the same mentor, in the same department, and more or less our topics were same when we did our PhD. We were basically working on macrophages, trying to activate them, and those macrophages, if you activate them, they can kill tumor cells. And in vitro, they do, in culture. And then I went on to do my postdoc in the same field, because I was hired based on my experience in macrophages. So went into the macrophage lab. And at the same time, they had cloned all these chemokines. These are small molecule cytokines which were important in recruiting these immune cells to the tumor site. And my project, which I will talk during the discussion group, what experience I had. But all in all, I can tell you is that when you bring these cells to the tumor, or when you see these cells within the tumor, most often they are not inhibiting tumor growth, but they are promoting tumor growth. So what we had in vitro was not actually translatable in vivo, unless really you go and manipulate very wisely and activate them appropriately, then you can see them. So that was my experience and then I continued on to same work, and still I'm working in the same thing, chemokine receptors, immune cell recruitment in tumors, and still I don't have answer to what exactly they do. So 25 years later, I'm still confused. So what I'm going to do, this morning and in next 4 or 5 days, trying to introduce you to some of the basic concepts and some of the hot areas, in immunotherapy and individualized vaccines, tumor vaccines, and then we will end up with having more and more discussion, and based on your question, we can deviate from wherever we want to go. This is a very wide open field, very hot area. How many of you had immunology before? Can you raise your hand? Wow, most of them. Okay, that's good. So I don't have to go in and introduce you immune system. And how many of you had cancer biology before? Few of you. So I think we may have to spend some time on cancer biology, and I think I can do it in the afternoon, 1 hour. I will try to give you some basic understanding of tumor progression and metastasis, and we can continue from there. So the objective of this whole session basically will be, try to give some understanding about immune surveillance, because as you all know, that immune system is our basic defense mechanism. And without its, if it is really working well, cancers or any other diseases will never happen. And failure is basically leads to all kinds of disease, including cancer. Inflammation is key. It is a key element in not just cancer, but many other pathological conditions, heart disease, Alzheimer, and you name it. So inflammation is very essential and important in immunology, because without inflammation, you don't have immune response. And chronic inflammation leads to all these pathological conditions. Immunological escape, these tumors basically, they do escape. And that's how they develop the tumors. Anti-tumor immune response, we will talk about what are the different cell types, basic mechanism, and at the end, we will discuss more about immunotherapy, because it is a multi-billion-dollar industry. If you turn on the evening news, you cannot miss an advertisement about all these therapies, claiming to cure cancer. So I think it's a huge, huge market. So we'll discuss about that. It's a hot area. Ultimate question is basically, whether immune response is effective against tumor or not. You will have that answer, and is it worth continuing developing new immunotherapeutic protocols? So I hope that, through our discussion and presentation, you will have a basic understanding to become a judge or jury, to claim to that by Friday, okay? That's a very far-reaching, an ambitious claim. And what I'm going to cover in 5 days, actually we cover it in 3 weeks. I give the same lecture, to our graduate students, and it takes 3 weeks. So I think I've tried to simplify many of the things, but and if you have any question, please feel free to stop me at any time. So let's start with simple definitions. What is cancer? You all know that cancer is an abnormal and uncontrolled growth of the cells. And think about cancer as asocial elements, in our society. So our body is like a social structure. There are rules to follow. Cancer cells don't follow any rule. So that's basic, simple understanding about cancer. And tumor immunology is basically study of the host response against cancer cells, including anti-tumor effect and what are kinds of different interactions have. So why cancer happens actually? Because if you look at, our body has a very, defined and very strong, complex mechanism to control these transforming events. If you look at a cellular level, a cell, most of the normal cells, whenever they get insulted, the first response they have is that is through intracellular mechanism, they will go and die, or they will go under senescence. So that's the first your control mechanism in cancer. So even if that mechanism fails, then there is another mechanism, is that cell which have already been transformed is within some environment, like matrix within that environment, other cells. So that's the second level of control. Those transformed cells will be there, but they will not progress. And third is if they happen to have some unique antigens presented, our immune system will kick in and they will try to clear them out. So these are the three basic mechanisms. That's why most of us will never develop cancer, and most of the healthy individuals never develop cancer. And the last part, where the immune cells get involved and try to control transform events, is what is the part of the tumor immunology. The first part is all cell biology, where all these apoptotic events, senescence, and all those things you study. And the host-tumor interaction, of course, is very important. When you look at the tumor, within the tumor, any tumor type you can think about it, somewhere between 20 to 80% of the cells are normal cells. What is the role of those normal cells and why they are there? The basic response is that they're supposed to control those cancer cells. But what happens is cancer cells bribe them, and that's how they cooperate and help the tumors to grow. So again, yes, go ahead.
When they enter the tumor, they are normal cells.
They are normal cells, but
do they remain normal after that?
No.
And we still call them normal? I guess not.
So they are not normal. Normal is genetically normal. So they don't have those mutations and other events, which happens in the tumor cells. So they can be manipulated very easily. And you can think about it again. I said the cancer is a social element. If a drug dealer wants to establish a market, what he will do? First, he will go and bribe who? Police, right? And all those, who are responsible for protecting you, they will try to go. And that's what the cancer cells usually do. They go and bribe all the other cells which are supposed to control cancer cells. So they basically exploit the homeostatic, mechanism supposed to control the tumor. So immunology, you all have had immunology before, and you are all familiar with understanding the immune system, which is a very complex network of specialized cells and organs, distinguishing between self and foreign molecule inside the body. Actually, this is a very key, important, distinguishing thing about immune system. So they are trained not to, attack, not to go against the self-antigen, but any foreign invasion, they are supposed to control. Like your defense system, you can think about your military. So basically, your immune system is like a military of your body. Basic function is protection from the pathogen, and malfunctioning of immune cells usually cause allergy, arthritis, and many other diseases, including Alzheimer's and cancer. One thing I want to emphasize here, that if you look at the immune malfunctioning, we have a lot of different diseases which are associated with immune functions overactivation. Cancer is the only one where immune system fails, So keep that in mind, that all the other immunological diseases which you can think about it, they are all related with abnormal activation of immune cells. Cancer immune cells basically are suppressed in many ways. Antigen, what is antigen? Against immune response? Is antigen. Any foreign particle that enters into body is antigen. You all agree? Okay, so what is antigen? It's very important to understand antigen before we get into anything immunology. Okay. Basic definition of an antigen. Okay, so I don't want to waste your time. So simple and basic, understanding what is antigen. Anything which can be recognized by T cell receptor or B cell receptor. You all know T cell receptor and B cell receptor. They are all unique for unique antigens, and we have plenty of them, different variety. So we can basically are equipped to encounter any and every antigen possible in the world, billions and billions of different variety. But basic definition of antigen is that particular molecule or foreign object has to be recognized by T cell receptor or B cell receptor. If they are not recognized, basically they are not defined as antigen. Okay. When you look at the immune response, yes.
Students usually, when I've been teaching immunology, two terms which they get confused is immunogen and antigen.
Oh, okay.
Many of 50% students
Yeah.
like
So all you need, remember what antigen, B cell receptor, T cell receptor. Okay, that's it. They have to be recognized by that. If anything else,
may or not,
yeah,
may or may not elicit an immune
exactly, may or may not elicit immune response. They're not immunogens, so they are antigens. Immune response basically is specific, and it has to be rapid and effective. Okay, so those are two specific characteristics of immune response. Rapid and specific. So again, system is basically like defined as interacting interdependent group of items as a unified whole. That's what immune system. You have a lot of different type of cells involved. They are organized into different organs, and those organs basically are of your lymphoid organs, lymphoid tissue, which you have all studied. And this is the simplest form of immune response we can give it, right from the high school. If you have any infection, your innate immune cells, because these cells they don't need to be trained. They are already, they can respond to these infections, come into picture, try to resolve, or pick up some of the antigens and go and present it to, T cells and B cells, and try to induce this something called adaptive immune response, which has to be specific for a specific antigen. Different cell types are involved in all these responses, and then you have clearance of the infectious agent or foreign object, and some of the memory remains. So next time if there is a same kind of infection, you have a very rapid response, because they don't have to go through the boot camp, which they go into these secondary lymphoid organs. So this is a very simplified version of immune system. Recognition of pathogens and elimination is two key events happen during immune response. Different cell types which are involved in this. This is again a very simplified version. They all are classified as one group called leukocytes. All the cell types which are part of your immune system, they will be called leukocytes. And within that you have lymphocytes, phagocytes, or other auxiliary cells, and other cells which can play also important role in immune response. This is a basic immunology 101. And the basic cell types, again, you are all familiar with B cells, T cells, macrophages, NK cells, eosinophils, neutrophils, and so on. All these cells basically do their job through different mediators. Either it could be soluble factors or could be cell-cell interactions where they can directly interact with the infected cells and take care of them. So this basically a very compartmentalized system where you have different cell types involved and different factors get into it. There is two different areas and two different sections or two different arms of immune response. I keep hitting that. One is your innate immune response, which is rapid. As I said, it does not needs to be antigen trained. And the other one is your adaptive immune response, which is a slow response. Different cell types which are important in innate immune response, including your macrophages, natural killer cells, neutrophils, eosinophils, basophils, dendritic cells, mast cells, and your granulocyte pool. In your adaptive immune response, you have two cell types which you have to remember, your B cells and T cells, which basically are the main force, in that. And there is a big overlap between these innate immune response and adaptive immune response. Some of the cells fall into this, this gray zone. One key element is that these things are happening in continuation or in dynamics. That early on you have innate immune response where they try to clear the infection or injury. Those antigens which are released during that process can be picked up by antigen-presenting cells, and those cells can go to secondary lymphoid organ and then present it, and activate the B cell or T cell or adaptive immune response. So this is a basic global view about the overall immune response which we have in general, not against tumor. This is a general immune response which we are talking. And most of the cells which are involved in immune system are mostly all derived from one cell type. It's called hematopoietic stem cells. In our adult, where it resides, where do you find the hematopoietic stem cells in adult human? Bone marrow. So mostly it is in bone marrow or fetal liver. And these cells, they get differentiated into different progenitors, and then they can give rise to all different kinds of cells in the immune system, which gets into the circulation. Many of these cells, once they cross into the tissue, they become different tissue inhabitant cells like tissue macrophages, mast cells, granulocytes, or antigen-presenting lymphoid cells. And there is a whole cycle goes on, like if you have infection, these cells cross into the tissue, then go back to the circulation, and they go back to the different secondary lymphoid tissue and do their job. So there is a very continuous pool of these cells getting produced almost every day, depending on the half-life of these cells. Most frequent ones in this category will be your granulocytes. How many granulocytes are produced every day? Give me a number. In adult, normal, healthy adult, how many neutrophils are produced every day? The half-life of neutrophils is now supposed to be 4 or 5 days. 24 hours it was thought to be earlier. At least, actually, in culture it is just 24 hours, but in human body it's 4 to 5 days. But how many neutrophils are produced? Million, thousand, billion? 3 numbers. Billion, million, thousands. Billion? Million? I think you will be immunosuppressed. Billions of them are produced every day almost, actually. And if something happens to hematopoietic stem cells, what will happen? If the person will be immunosuppressed, right? And most of our chemotherapies, what do? So chemotherapies are, most of the majority of the chemotherapies are common, radiotherapies. They target dividing cells, So whenever you are giving these treatments, first thing you will see is that they're not just targeting the tumor cells, but they are also targeting all the dividing cells. And one of the most rapidly and usually everyday dividing cells is your hematopoietic stem cell and all the progenitor cells. So you will see the common feature in these patients, they become myeloablative. That's one thing you see. The granulocyte number goes down, that's one feature. And once that number goes down, they become very susceptible to all kinds of infection. So there will be a question on that too, actually. The chemotherapy, radiotherapy causing myeloablation, and then what happens, and which cell types comes into the picture. Lymphoid tissues are very important, not just for the immune function, but I will talk about it in tumor progression also. These immune cells, usually, like your defense system, you cannot let them loose, because these are all very armed and dangerous. 16-year-old kid with M13 machine gun. Think about it. You don't want to be freely roaming on the street. So you have to organize them in some sort of barracks, and some sort of depot. And your lymphoid tissues or secondary lymphoid tissues basically play that role, where they get the right signal where to go and what to do. So that's very important, and that's why these lymphoid tissues are important. They usually comprises of your lymphocytes, macrophages, antigen-presenting cells, and other epithelial cells, which are not just bystander. They also play a major role in that response. It is arranged in directly encapsulated or loose, very devoid lymphoid tissue. Encapsulated secondary lymphoid tissue, the best example is all those lymph nodes and spleen will be the best example. And the diffuse lymphoid tissue the best example will be all those which are associated in your alimentary canal, MALT, the mucosa-associated lymphoid tissues. Cells are different in those two compartments, and the responses are different too. Primary lymphoid tissues actually are the ones where the lymphocytes are produced. Give me the example of a primary lymphoid tissue. We talked about it 2 minutes ago. Bone marrow is one. Thymus is another one, yeah. Although cells are not produced, but that's where they get trained, go through the boot camp. Secondary lymphoid tissues, these are critical for lymphocyte antigen-presenting cells interaction and important for the immune function. And here is a very simplified version of different primary lymphoid tissues and different secondary lymphoid tissues. Depending on the organ, they are named differently, but all you need to remember clearly is lymph node, spleen, and other lymphoid tissues. Bone marrow is actually listed in both sides of this. Any reason for that? You see that bone marrow is primary lymphoid tissue and also secondary lymphoid tissue. Textbook, it's not any science here. Why? Of course, we all know why it is in primary tissue, because that's where your hematopoietic stem cells are coming. Why it is listed also in secondary lymphoid tissue. What happens to B cells, actually, yes. Where do they go once they have all the signals and all the interaction? What antigen they have? They go back to bone marrow again. So they also work as a secondary lymphoid tissue. So here is your first test. All the cellular elements of blood, including lymphocytes of immune system, arise from hematopoietic stem cells in the bone marrow. So what is this cell? Based on your basic immunology, what you know. You just said hematopoietic stem cell. Everything is coming from there. Okay, I'm not going to go through all the other box. What is this one? This is macrophages. And what is this? Dendritic cells. And what is this? This is granular. This monocytes. So what's the difference between monocytes and macrophages? What's the difference between monocyte and... No consensus? Forget about the shape, So they are same cell actually. In circulation, they are monocytes. Once they get into tissue, they become macrophages, right? More or less, they have similar function. Maybe macrophages are little advanced, because they have to do some tissue-specific activities. And there are a variety of different type of macrophages, depending on which organ you are working on. They are named differently also. So what is this cell? This is coming from your lymphoid progenitors. This is B cell, T cell, B cell. What is this? Plasma cells. These are antibody producing, cells which are plasma cells. Okay, test is over. Let's move on to the next. So this is our basic immunology. I didn't go into the specific antigen, and CD as those T cell antigen presenting cells interactions, because we will have a whole lecture on that. Okay, so let's get into the tumor immunology. And first basic concept in tumor immunology is our immune surveillance. Which basically what it is saying is that our immune system prevents the development of most tumors, which the whole concept started sometimes in early 19th century, and it's still, people are trying to show experimentally that it works. Our host immune system is constantly surveying our body for abnormal cells and kill majority of the tumor cells before they become clinically apparent tumor. And you all can understand how they do it, that if you have any transformed cells, those transformed cells will start, depending on the transforming event, carcinogen or virus or whatever agent which is causing the transformation, or even the simple, genetic events which causes the transformation will cause the expression of specific unique antigens, and those antigens can be recognized as foreign, not the self-antigen, and those cells can be cleared out by which cells? T cell, B cell, macrophages. Antigen, it is antigen specific, so no macrophages. So which cell will clear the transformed cells if they are expressing unique antigen? T cell, I can give you that much hint. Which T cell? Cytotoxic T cells. Which one is cytotoxic? CD4 or CD8? CD8 positive cytotoxic T cell. So now I think class is over. We are all done. I think you know and more about tumor immunology than I know. So done. CD8 positive T cells, cytotoxic T cells are important. And that's what actually they are major player in immune surveillance. So as I said, the concept of originally started in early 1900 by Paul Ehrlich, and basically what it was suggesting is that cancer could occur at an incredible frequency if our host defense system is not working, and that you can see it in those individuals who are immunosuppressed, are immunodeficient. The incidence of cancer is, in normal population we are talking about 1 in 2 or 1 in 3. And in those population, we are talking about 80% of the individuals who are suffering, who are AIDS patient actually. The best example I can do. AIDS patient, if they live longer, they will develop cancer. So I think immune system is very important. Here is a normal feature, normal cells. Usually they have self-antigens which cannot be recognized by our CD8 positive cytotoxic T cells. And there is no, it's a self-tolerance, whatever you call it. In a malignant cells, these malignant cells start expressing unique antigen, and these antigens can be recognized by your CD8 positive cytotoxic T cells, and which can clear them. And that's what happens in most cases, if immune surveillance is effectively working. I keep saying here is I have not said so far. It's class 1 MHC is listed here. All MHC, major histocompatibility complex, and these they are very important for antigen presentation. Okay, class 1 MHC here it's listed. Which cell type expresses class 1? There are two types, class 1 and class 2. Which one is expressed, which cell types express class 1? I'm not just going to come and lecture you, okay? I'm going to ask questions. You have to give the answer, I think. All cells in our body, they express, class 1 MHC molecule. That's why, if any cell type which is transformed are having, those unique antigens presented, they are always associated with class 1 and your CD8 positive T cells. And another thing is that your CD8 positive T cells only recognizes those antigens which are presented in association with class 1 molecule. So which cell types express class 2? APC. Somebody said APC. Antigen presenting cells. All antigen presenting cells, professional antigen presenting cells, they express class 2. Okay? And the example of those antigen presenting cells are your dendritic cells will come into the first category, macrophages, and B cells. Okay? Those 3 cell types, they can express MHC class 2. Okay? These antigens, again, class 1 and class 2, depending on where these antigens are coming from. Okay? If it is coming from intracellular, it will be presented in association with class 1 or class 2? Class 1. If it is coming from external sources, they will be taken up by your antigen presenting cells, getting processed, and will be presented in association with the class 2. So that's very basic, concept in immunity. So in tumor immunology, both class 1 and class 2 are important, but the cell type which is important is CD8 positive T cells, and most of the time, all the time, they recognize the antigens when they are associated with class 1 MHC. And what are the different examples and the evidence for immune surveillance? Other than that, not everybody gets cancer. Many tumors contain lymphoid infiltrate, which you can see in many tumor types. There are spontaneous regression of tumor in human as well as in animal models. You can see that happening. Tumor incidence is high in neonatal and aged or immunocompromised individuals where the immune system is compromised. You can see the incidence of tumor is very high. Again, immune surveillance, it depends on the type of tumor-inducing agent. In some cases, it's very strong and effective. In some cases, it is not. If it is induced by some viral agents or chemical-induced tumors, in these cases, our immune surveillance is very, very good, actually. So I think if you are healthy, your immune system is working well. These exposures to different viruses and chemicals, probably you can take care of. But if it is some events like genetic mutations and other spontaneous genetic changes which happens, and then the cells become transformed, in that event, our immune surveillance is a little weak, actually, because most often, these cells, they are not having enough or sufficient or unique antigens which can be recognized by your immune cells. Most often, those antigens which are presented, they look like and they act like, your self-antigen. I kept saying antigen, antigen, antigen many times. One thing, again, it's very basic you need to understand is that there is no tumor-specific antigen, okay? You have tumor-associated antigens. So that's, most of the antigens which have been targeted you can see that happening in tumors. Those are, I will classify them as tumor-associated, not tumor-specific, because those antigens can also be expressed by the normal cells somewhere and at some occasion. There are some other non-supportive evidence, where that can be challenged. Many lethal cancer arise in immunocompetent individuals, which we see, the normal healthy people getting cancer. And we cannot explain, why their immune system didn't work. And there are many things we can explain, but none of them are satisfactory. Immune response against solid tumor is very weak, and this is one area where a lot of work is going on trying to make them more immunogenic or hot before you can use different immunotherapy. And the immunodeficient individuals often develop hematological malignancy, which is still not clear why, that in those individuals, I gave you the example of AIDS patient. If you look in the incidence of different tumors in those AIDS patients, 80% of them are hematological malignancies, and these malignancies basically often they develop because of the abnormal cell division in your hematopoietic stem cell and aberrant activation of your B cells, T cells, or macrophages, or myeloid cells in general. So still, it is not clear why. So still it's debatable, and which is good for science, actually, that the work goes on. But if you look at overall very generalized anti-tumor immune response, you have tumor cells, which can be targeted by many different cell types, cells of your innate immune system, which can try to target and kill those tumor cells, or your cells of adaptive immune response through the different production of cytokines or antibodies can target the tumor cells. This is a very simplified version of anti-tumor immune response. And now we can get into some little experimental detail, what are the evidence of anti-tumor response. And this concept very early on in 1950s, most of people, very basic, immunology they were trying to do, they will use these, irradiated tumor cells, and most of these tumor cells were derived by carcinogen-induced, tumor models. So they were very immunogenic. So they will irradiate, they will introduce these irradiated tumor cells into mice, and what will happen, that these mice will develop immune response against those tumor cells. And if you give that tumor cells to the mice, those which were immunized with those mice will be protected. And if you give it to the naive mice which are not immunized with these mice will develop tumors. This was shown in the chemically induced tumor models. It works, and it works through your basic immunological response. Like if you introduce anything foreign, you develop immune response against that specific pathogen. So this was actually basic first evidence that show that tumor-specific immune response happens. The other examples where where that inject the tumor cells into the mice, and then they will resect the tumors basically, and hope is that this tumor, these mice will have some immune response against that tumor which was introduced. And again, if these mice were re-challenged with the same tumor, it will be protected. But if you take the naive mice and inject the same tumor, those mice will develop tumor. Again, this is carrying those immunity which they develop early on during the first phase of their, it's like a vaccination. Vaccinated these mice, they are protected from the re-challenge of the tumor, and they become tumor-free. Again, the big in bracket thing is that most of these studies were done with the tumor models which were highly immunogenic, and all were done by the chemically carcinogen-induced tumors. And at that time, the basic finding was that if you vaccinate these mice, prior on before challenging with the live tumors, you can protect, these animals. If you can do otherwise, that you challenge them and then you vaccinate them, they will not be protected, which was, expected that's how it will work. All these things, your basic immunology and immune response, what you can expect if you introduce any foreign antigen into the system, your immune response can be. And this was okay with these type of tumor, but in reality, it doesn't happen. Even in animal model now, we don't see that. If we use, other tumor system, we cannot see the same phenomenon. But if you use those MCA-induced or other carcinogen-induced tumors, you can see the similar phenomenon. All in all, what it is telling is that you can generate antigen-specific immune response against tumor if you have the defined tumor antigen, or if your tumor is highly immunogenic. Again, now we will get into what are the different cells which are important against the tumors. Your innate immune response, basic players, what you have, natural killer cells, PMNs, monocyte, macrophage, and killer cells, they all play a different role in controlling the tumor, cells. They can kill them through the different mechanism. NK cell, basically their main mechanism is through production of these factors called granzyme and porphyrins. They basically can get engaged. They can recognize these tumor cells. How NK cells recognize tumor cells? Anybody has any thought? CD8 positive T cell recognizes tumor cells by antigens which are associated with class 1 MSC. How NK cells recognize? Natural killer cells. That's your homework, okay? You can tell us about tomorrow or day after, okay? Basic, I can give you the clues, that all the in this case, basically, those tumor cells, it's not associated with MSC molecule, okay? And there are some activating signals which gets activated in NK cells, okay? Those are the two clues I gave you. You can get the answer. But once they recognize these tumor cells, they produce these death factors, your phospholigand, another death mechanism, granzyme and porphyrins. They can create holes in the tumor. They can induce apoptosis or necrosis or necroptosis or pyroapoptosis. No matter what, they are dead, okay, whichever pathway they go through. Another mechanism which NK cells use to kill the tumor cells is through this called antibody-dependent cellular cytotoxicity. And in this case, basically what it is that, these antibodies which can be produced by the specific antigens which are on the tumor cells, those antibodies will recognize those antigens. Not in MSC class 1, they can recognize, as it is, because these are antibodies. And once these antibodies are bound to these antigens, antibody is a bispecific molecule, In one portion of the antibody is for recognizing the antigen, and the other portion of the antibody is for what? To do the job, The job is to clear the cells, kill the cells, or do other things. So basically, these bound antigen-antibody complex can be recognized by Fc receptor expressing cells, and NK cells, they do express Fc receptor, okay? So these cells will be recognized by NK cells through this complex, and this phenomenon is called antibody-dependent cellular cytotoxicity. Okay, and other cell types can do the similar thing, and other cell types which have this ATCC mechanism will be, what other cells express Fc receptor? You know what is Fc, right? Your antibodies have FAB and Fc portion, right? So FAB is your antigen recognizing domain, and Fc is which can go and bind to the cells which express Fc receptor. Which cells express Fc receptor? NK cell. What other cell type? Neutrophils, macrophages. Those are the other two major cell types which express Fc receptor. Adaptive immune response against tumor. So in innate immune response, I just mentioned NK cells. I left the macrophages and the other cell types, which we will come back and cover it later. In adaptive immune response against tumor, it is against the tumor-associated antigen. Cells which are involved in this process will be your T helper cells, which are mainly your CD4 positive T cells, and cytotoxic T cells, which are predominantly and mainly CD8 positive T cells. CD8 positive T cells, they recognize antigen in association with MSC class 1. T helper cells, basically, they are important for mediating these immune response through production of different immunomodulatory cytokines. One of the key events is that these processes have to be controlled and well characterized, and the responses has to be optimal. And in many cases, when in most of the therapies which are basically utilizing these cells to treat cancer, this is the part where we are failing and causes the toxicity which are associated with these treatments, because we still don't know what is optimal. How much cytokines, how much cellular activation is critical to remove these cells, because this is happening in a live individuals. Most often, most of the immunotherapies which we have, they are all associated with severe toxicity. The cytotoxic T cells, which are important in clearing these tumor cells, these, again, is simplified example is CD8 positive T cells. They will have these antigens presented to them in MSC class 1 fashion in association. Your T cell receptor will recognize these antigen, you start producing these perforins, granzymes, or phospholigands, and they can kill, these, tumor cells which are expressing that antigen. So if you look at these two cartoons of NK cell-mediated killing and cytotoxic T cell-mediated killing, most of the effector molecules are same, your phospholigand, granzyme, perforin. Only difference is one is happening in context with your antigen, MSC class 1 molecule, and the other one happens in the absence of MSC class 1 molecule. We have a very effective immune response, but as I said, in most cases it fails, to have effective immune response against the tumor or antigen-specific T cell generation against the tumor. And most of the defects, which I'm going to show you in this slide and the next one, is all associated with the functions which are normal for these immune cells to do it. Defects in T cells. First one which comes into the mind is they are not enough. When you look at the tumor cells, hardly you see, T cells, and one of the basic, criteria right now, pathological finding is that if you can find more, T cells within the tumor, prognosis is pretty good. Okay, so you don't have to go do any functional analysis, or even if you do some kind of treatment, if you can see increased in the frequency of simple CD3 positive T cells, you can expect that individual to survive longer compared to those where you don't see the T cell infiltration. There are not enough cells within those tumors, T cells. Induction of energy. So energy basically happens where they can recognize the antigen, but they cannot respond, and the response is lacking because they lack other signals which are important for the, those T cells to become effective. Downregulation of TCR receptor zeta chain. This is another important basic understanding in immunology. When you look at the T cell receptor complex, this T cell receptor zeta chain is the one which is important in downstream signaling. And if it is missing or downregulated, even if they have everything, all the recognition and secondary, activation, they will not be functional. Suppression or help from T cells and different inhibitory cytokines, failure to develop and retain memory. There are defects in antigen-presenting cells and antigen presentation, starting from a cellular adhesion to MSC molecule expression, defective antigen processing and transport, absence of antigen. This is another important process which tumor cells utilize. They don't have any antigen actually which can be recognized by your T cells or can be presented by antigen-presenting cells. Modification of viral or tumor peptide antigen. Absence of co-stimulatory molecules, this is the secondary signal which I was talking about in terms of energy, that in order for these antigen-presenting cells and T cells to interact properly, not only the MSC and TCR interaction is important, but another whole class of co-stimulatory molecules get involved, and they are important for effective immune response. Abnormal activation and checkpoint inhibition. This is another, where last year they got the Nobel Prize. Jim Allison, and company got the Nobel Prize for checkpoint inhibition. The difference between these absence of co-stimulatory and checkpoint inhibition is that in this, basically you are looking into the antigen recognition by antigen-presenting cells and T cell. Checkpoint inhibition is more or less like, senescence, that T cells, they already have everything they need to, but they don't act. So this is the basic difference between these two. And both of them has been targeted, or many actually, not both of them. Many of these features which I'm listing here has been targeted as a part of immunotherapy, trying to fix those problems, and those are all part of our immunotherapeutic protocols currently in preclinical or different clinical stages. Different in microenvironment, defects in microenvironment where there are a lot of immune inhibitory cytokines which are produced by the tumor cells or the cells surrounding the tumor. Privileged sanctuary, some areas are very, privileged. What are the immune privileged sites we have? We have certain organs which are characterized as immune privileged. It's very protective organ and very important. Just one example. Use your brain. It's not got you question, okay? I'm just so what is immune privileged site? What will be characterized as immune privileged site? Where immune cells actually don't get in and out very easily, or don't even get in and out. Huh? Brain. Brain is one. Is there any other one? Think about it, okay? You can Google it, okay? Dr. Google will answer you. I'm not going to give. Brain is definitely immune privileged organ, okay? Latency is another important issue in the tumor, which we will talk about when we will go into immune, editing and immune surveillance. So all in all, there are a lot of defects which are mostly associated with the different cell types or in the environment, which are causing these failure or immunological escape. So the immunological escape in tumor immunology is the central paradox, that despite the fact that we have such a intricate system to clear these tumor cells, still these tumors develop, and they develop because all the defects which happens in the tumors, tumor response, which I listed before, and many more, which are still unexplored. The fact is that there is a very fine balance which goes on when you look in context with the tumor, which favors either tumor growth or tumor destruction. And in most cases, actually, tumor destruction wins. And again, I'm saying that because most people don't get cancer. In some incidences, they become and help tumor growth. And this is one of the area where, actually, if we can understand how the tumor cells escape our immune response, that can be exploited, and that has been exploited to develop different immunotherapeutic protocols. So again, in terms of tumor escape, one of the things which comes into picture first, like I just said, there are very few immune cells within the tumor, developed tumor. We don't know much about developing tumor. Early on, you can see many more infiltrates, but as the tumor progresses and develops, but you see clinically evident tumor. At that stage, there are very few, immune cells or leukocytes within that tumor. Tumor cells really grow very fast, so the division, what is happening, that overwhelms, So I think here, all the cell things which I have listed here, which are associated with immunological escape, is overwhelming the system. So even if the response is perfect, it's not sufficient. And I will, if I have time, I will give you some of the actual clinical, examples where we have looked at the immune response, cytokine profiling, immune cell profiling in the cancer patient, who are going under treatment. And you can see that any cytokine or cell type you want to look, these patients have it. Actually, they produce more than normal people do, but still there is no response. These immune responses are totally, they don't work. Antigenic modulation, there is no specific antigen which can be targeted. Masking, antigen masking is another phenomenon where most of these tumor cells utilize different mucins to cover and mask the tumor antigens, which can be recognized to prevent recognition. Antigen shedding, actually this has been a challenge, but actually this has also been exploited in different therapeutic protocol now to profile the tumor or profile the patient, because these soluble antigens can also be recognized through different techniques, and you can basically tell it has been utilized as a biomarker. And best example I can give you is like CEA. In many GI tumors, they use CEA as one of the markers. CEA is nothing but an antigen, which are shedded by the tumors in the circulation, and which can be targeted using different immunotherapeutic protocols. Tolerance is defined where the immune response is there, but it doesn't work. Your antigen-presenting cells, they do their job. T cells are generated. But those T cells, they cannot function because of the suppressive environment or different cytokines which are produced there. So there is a failure of inducing effective cytotoxic T cell responses. There are genetic factors which have been shown to play an important role, and the most important one is this loss of MSC class 1 molecule. And you can understand that if the tumor cells don't have MSC class 1 molecule, they will not be able to present any antigen which can be recognized by the tumor cell, your CD8 positive T cells, and then you have tumors basically escaping. Different inhibitory factors, that can be cytokines or cell-associated molecules which basically suppress the cytotoxic T cell response. And above all, the tumor cells basically are the major producer of different inhibitory immune cytokines and immunoregulatory molecules, which basically are suppressing the immune response and allowing tumors to escape. So here is one simple cartoon showing that tumors which basically there is low immunogenicity, means no peptide MSC molecule, no adhesion molecule, no co-stimulatory molecule. Your T cell is all armed, but they don't know where to fire or where to act. In other case, you have these antigen which can be recognized by antibodies, but these antigens can get internalized by these antigen-antibody complexes, getting internalized and then getting degraded, and now these tumor cells cannot be recognized by your immune cells, CD8 positive T cells. Or in third situation, where everything is there, your cytotoxic T cells are there, they are getting right help from the CD4 positive T cells, but these tumor cells are producing tons of immunosuppressive cytokines, and if you have to pick one immunosuppressive cytokines in these patients, TGF-beta comes into picture. Okay, please remember that TGF-beta is one of the most important immunosuppressive molecule, and then you have tumor-induced immunosuppression. So at this, I will take 2 minutes break. Okay, and then we will get into, and if you need, also I think you need 2 minutes break too. We will get into the second part of this. Okay. Do you have any question before I give you? Yeah, we have 5 minutes. We can get you 5-minute break. Any question before break? No? Okay, I think. Yes? They will develop cancer, yeah. The immune system is compromised, so in those cases, any cellular transformation is happening will become full-blown cancer. And one of the things in these immunocompromised individuals developing hematological malignancy, the basic mechanism is very simple. When you look at B cell development or T cell development, if you recall, how B cell basically, gets out of your, bone marrow, where they get maturated, and what are the different events happening in T cell maturation and B cell maturation, you will find the answer. During the B cell maturation, as the B cells are, from the lymphoid progenitors, these cells basically go through the whole process of, recombination event, to decombine and generate these B cell receptor and T cell receptor. And anytime these recombination events are happening, these are very prone for abnormal events to happen, because these are genetic events. As they are recombining, they are creating normal productive recombinant events, and also abnormal non-productive events. And actually there are majority of those recombinations are non-productive, and they get cleared by your immune cells or through other apoptotic mechanism. But if your immune cells don't work, those cells which have non-productive recombination events which happen during T cell receptor or B cell receptor generation, they get accumulated and they become tumor. So first, there is no transmittable cancer. So cancer cannot be transmitted. Okay, cancer cannot be transmitted from one individual to another. Okay, The cancer which are associated with infection, right? That's what. So when I say transmittable, my understanding is that it gets transmitted from one individual to another through contact or through, some. Yeah. Yeah, so individuals who have H. pylori, they have a higher incidence. Individuals who are infected with HPV, they have a higher incidence of developing cervical cancer. Individuals who have HIV, they have a higher incidence. So they are susceptible to developing cancer, these type of different type of cancers, But I don't think those microorganisms are transmitting cancer from one individual to another. Yeah. Yeah. Yeah, I don't think, so cancer in general, if you think about it, that why people get cancer, so there is no answer, If you think about hereditary factors, less than 5% of the cancer are associated with a specific, genetic, abnormality, which goes from generation to generation, less than 5%. 95% of the cancer is sporadic in nature. And even in that, of course, we have etiological factors like HPV, H. pylori, and HIV or other factors which we can associate it. So those are like etiological factors, all derived from epidemiological data that those individuals which are exposed to those factors are more susceptible. But in reality, that even if you are exposed to those factors, it doesn't mean that you will develop cancer. My perception will be that it's a bad luck, that somebody develops cancer. In more or less, you cannot, tell that. Yeah, of course, some smokers have high incidence of lung cancer, but you will find smokers who will never develop any cancer, So I think how we can argue, but I will say that it's bad luck. Of course, there are factors which are associated with it, and actually they have also discovered the causative elements within those etiological factors. But yeah, tobacco is one of them. But it doesn't mean that they always cause cancer. So, but in general, I will be very, concerned if I call a cancer as a transmittable disease. So you can argue against, okay? This is what science is all about. Yeah. Mm-hmm. Yeah. But actually, these cancers where we have identified and which are associated with a specific infectious agents, they have been very successful in preventing them, like with HPV vaccination, that has been really a wonder, Yeah, yeah. And HCV can be another one, which they can do it. So I think this is a, area where if really we can identify these, type of thing, we can prevent. Yes.
Sir, can immunosuppression because of transplants trigger any like?
Yeah, so those individuals who undergo transplant, they are always under this immunosuppressive regimen, and those individuals have higher incidence of cancer. Yeah. Not just transplant patient, also those people who are getting treatment for arthritis. Most of those treatments are immunosuppressive. Or, most common one is use of steroids. Those individuals who are on, chronic treatment using steroids, they have higher incidence of cancer. Huh? I will. It seems like even computer needed a rest. Oh, God. अपने आप बंद हो गया था क्या? Okay, welcome back. So now we can start a simple discussion on talking about cancer immunoediting. This is one of the, item which you hear more often in cancer immunology, cancer editing. And basic concept here is that as the cancer is evolving, when you see early on, is that your immune cells are very much actively involved, and most often they try to prevent that cancer ever to develop. So they are tumor inhibitory. But as the tumor progresses, these immune cells basically they don't function, and then this becomes like a tumor stimulating, a tumor supporting event. So this suppressing and supporting event is the whole process that defined as immune editing. And in this basically what you will see that if you immunoprofile these early tumors, middle tumors and late tumors, you will find a different profile of immune cells and cytokines or factors, whichever you are looking at, they will be different. And actually looking at the profile, you can tell that this is going to be tumor suppressive immune response, a tumor promoting response. And that's what all encompasses the cancer immunoediting. And basically there are 3 different steps which happens during this editing process. Early on, as I said, that when you look at as the cancer is progressing, majority of these cells will be so-called non-malignant. And non-malignant in terms is that these tumors don't have potential to metastasize to different organs. And usually they remain like homogeneous in nature. They are not very heterotypic, not very well diverse. And that can be induced by different factors like your carcinogens, radiation, viruses, chronic inflammation, or other genetic mutations. These tumors, when you look at their immune profiling, you will see these tumors, you will be your, very immunogenic response. You will see a lot of different immune cells infiltrate these tumors. And these cells include your CD4 positive T cells, 8 NK T cells, NK cells, and dendritic cells, gamma delta T cells, macrophages. So every cell type you can think about it, which plays role in immune response. They are all here. They have the right cytokines which are produced. They have the right molecules which are expressed. And at the end, what you will have is that this tumor will be eliminated. And this is what you see, what happens in process of immune surveillance, that these events happen and these cancer cells will be eliminated because of the response which you have, which will be effective, which is mediated by a variety of different cell types. As these tumors basically evolve, what happens is that, many signals still be unclear about is what causes tumors to progress further. These tumors enter into phase where the tumor cells in itself try to become more heterogeneous, and when it becomes very heterogeneous in response, the different kind of immune profile you will see, where you will have a very fine balance, where your immune system still is active and effective, but the tumor cells are not getting eliminated, because some tumor cells which are very susceptible, which can be eliminated, they get eliminated, and they call them as a highly immunogenic transformed cells, and that can still be controlled and remain in a, although transformed, but not progressing tumor, okay? And this will also still be in some sort of benign condition and not have still developed the malignant potential as the potential to metastasize to distant organ. And the cell types and functions which you will see immune profile, if you profile them, you can find a lot of CD8 positive T cells, NK cells, NKT cells. In general, you will find a lot of T cells, dendritic cells infiltrating those tumors, and cytokines which are produced, and most of them are immune enhancing cytokines like IL-12, interferon gamma, IL-23, or IL-17, and so on. And these dormancy exists for a long time, depending on the tumor type. Certain tumors, the dormancy could be months to years. If you think about cancer doesn't develop overnight. Most of the solid tumors, when we think about it, they develop somewhere between 5 to 20 years. It takes that much time. Best example I can give you is pancreatic cancer, where we work on. The latency period for pancreatic cancer is 20 years. Almost, you can think about it, that when that transformation event happen and when you can have a detectable tumor or the clinically apparent tumor. So these tumors remain dormant, because your immune system is still active and effective, but not able to eliminate, those transformed cells. And something happens here at this stage. It's very unclear, what causes these, dormant tumors to get reactivated and move into this progressive phase. And majority of the evidence so far suggests is that the main culprit in driving these dormant tumors to progressive tumor is immune response. Something happens during that event when our immune response becomes weaker, and these tumors basically start to progress. And at that stage, these tumors become so-called, in general terms, poorly pro-immunogenic, means the immune response against these tumors are very weak. They start to produce a lot of different immunosuppressive functions, different immunosuppressive cell types. They infiltrate them, tumor-associated macrophages, myeloid-derived suppressor cells, T-regulatory cells. Different surface molecular expression profile totally changes in those cells, like all these HLA markers or CD molecules. I'm not going to go through this list, but in general, what happens is that many of these immunosuppressive cells, cytokines, or factors get upregulated, and immunoinhibitory factors predominate these tumors, and this basically creates a more progressive tumors, become more heterogeneous, and becomes more malignant and metastasize to distant organ. So this is a process, going from tumor which was basically, highly immunogenic, and can be eliminated by your immune response, holding in more like in a dormant phase and driving it to the progressive phase. That all together comes into the picture of this whole called immunoediting. They are basically your immune system is editing the tumor cells, and that's what immune editing cancer is all about. That can be exploited very effectively in driving backward. Basically, the immunotherapies can be designed to, target these immunosuppressive microenvironment, make these tumors more like dormant tumors, or bring these cells and factors which can basically eliminate these tumors, and that's the goal of your immunotherapy. Current immunotherapy is reverting these progressive tumor back to benign stage, or altogether eliminating them. One of the key elements I can tell you, which is a side note in that, is that with the concept bringing back of the immune surveillance, these events are happening more frequent than we can think about it. It happens, every day, I will say. If you go and look at those individuals who die after age of 75, not because of cancer, for unrelated, events. And if you go and do their autopsy, you can find a lot of dormant tumors. They had those nodules or those dormant tumors, but they never develop, full-blown malignancy or detectable malignancy, and that's very evident, in today. So, of course, you all know that cancer is very much related with aging. So as you age, the incidence of cancer is higher and higher, and majority of the tumors are cancer of old age. In India, it's a different profile for many different tumor types, but in general, worldwide, cancer incidence is higher in older individuals. And what happens when you get old? You become more active. What happens to your immune response? It goes down, right? You become basically more and more immunosuppressed. So I think your immunity and immune response, again, by this, far-reaching evidence can suggest that they control the tumor. Yeah. Mm-hmm. Yeah, thymic atrophy. But I think by the time thymus gets into this atrophy, at adolescence, we have all kinds of T cells, with T cell repertoire. Yeah, that changes the T cell repertoire, So I think as we age, our T cell repertoire becomes, right now it's very diverse. It becomes more clonal, so it can do better job with certain, antigen, but cannot do certain job with others. So yeah, T cell repertoire will be also changes. So my pointer doesn't move. Okay, I can go with this. Even this doesn't move. I don't know what I'm in edit mode or what. And now it's what happened? It goes to sleep. I think I should try flash drive late. The next one. Yeah. This computer is going in sleep. Yeah. Sorry, it's a technical problem. Yeah, 5 minutes sleep mode. Yeah, good. Okay, we are back. So sorry for interruption. So this is all again rewording what we talked about. The process proceeds through 3 phases: elimination, equilibrium, and escape. If the tumor destruction occurs, the elimination constitutes the total removal or eradication. In most cases, the growth of these tumors very much depends on cellular immunogenicity, which can be edited by our adaptive immune system. And in these edited tumors can escape, through the different immunosuppressive pathways, which can be exploited for our immunotherapeutic, it has been exploited to develop different immunotherapeutic strategies. So this is just rewording. So all of these are targeted against tumor antigen, okay? And again, you all know about the antigen. We have basically introduced, concept of antigen. There are no tumor-specific antigen. There are a lot of tumor-associated antigens which have been identified. That can be dependent on the, the events which causes the tumor to develop. Oncogenic products have been, used. Embryonic proteins also can start expressing during these process of transformation. Viral proteins, a lot of different viral proteins have been identified as a tumor antigens. Tissue-specific antigens that has been exploited to develop different tumor vaccines. Mutated tumor suppressor protein like p53. Almost half of the solid tumors which we know today, they all have this mutation in p53 and idiotypic isotype. And we get into detail about different tumor antigens, because that is important in immunotherapy and understanding tumor immunology. As you all know, that your normal cells, basically they are expressing these self-peptides which are bound to your MSC class 1 molecule, and these your T cells will not recognize these cells because they have these training not to recognize the self antigen. But if there is anything happens where these self antigens get mutated and they can see something different, which gets recognized by these your CD8 positive T cells, and that cell becomes tumor and can be eliminated, or they can get modified, and that can be again recognized by your CD8 positive T cells, which can be and which can clear the tumor cells in itself. You all know how that self tolerance happens. It all happens in thymus. Okay, it is a very important process during your T cell development, and during that process there are two specific events happen as the T cells are developing. Negative selection and positive selection. You all know about that. That happens in thymus. So negative selection is against what? So, yes. So one of the thing is that these T cells should not recognize the self antigen, right? So that's one selection. Yeah, where they get trained not to recognize the self antigen. What other event happens, during antigen recognition when they are interacting? These antigens are presented with what? In association with MHC molecule. Okay, so those two events happen actually. So one is against the antigen, and another is MHC. So which one is positive? Which one is negative selection? They are getting selected for a specific, cell antigen, and they are getting selected for MHC molecule because there is a dual recognition happens during T cell receptor interaction. So which one happens for positive selection, or which one is negative? Self antigen is negative selection. So they should not recognize the self antigen. So those cells which don't recognize your self antigen, those who recognize your self antigen, they get eliminated. Yeah, and then they go through the MHC recognition, which is positive, where they have to recognize the self antigen. Sorry, self MHC. In order to. So that. All in all, this is a very complex system, okay? But you need to understand that, then it becomes easier. Other events where, the normal cells, they can get reactivated, some of the germ cell genes, some of those developmental genes which we were talking about, like in melanoma, MAGE3, and then those can be recognized, by the immune cells. There is overexpression of certain type of genes, and most of these are oncogene-associated products, which can be, as recognized by your T cells, as they evolve. So there are a variety of antigens which can become target for your CD8 positive T cells, and here is a very simplified list, and this is just giving you a flavor of different potential, antigens which can be targeted at the origin. That can be tumor-specific mutated oncogene, germ cells, differentiation antigen, abnormal gene expression, abnormal post-translational modification, or oncoproteins. And many of these which are antigens which are listed here have been targeted, to develop strategies, to develop vaccines against them, or to develop other strategies, and the different tumor types which they have been associated with. One other thing which you will find in this list is, in general, when you look at the immunotherapeutic, success, melanoma comes as number one. All these, checkpoint inhibitors, CTLA4 antibody, these lymphokine activated killer cells, any of those immunotherapeutic strategies which we, come to clinic, many of them have been successful against melanoma, because melanoma is a very immunogenic tumor. That's a very well established phenomenon. There are many gene antigens which have been identified and can be targeted. But on the other hand, there are other targets which have been exploited, for different tumor types, and this list is still keep growing. And I'm not going to go through each one of them, but just giving you an example that many of these classes of antigen can be exploited as a tumor antigen and can be used to develop strategies to develop vaccine, or can be targeted using other approaches in immunotherapy. One of the thing which is very common cancer in India, the cervical cancer, and most of them are associated with HPV virus. So these antigens basically are there, and with current vaccination thing going on, I think it's very going rapidly in India. Our hope is that by in 10 20 years from now, cervical cancer will not be in top 10 cancer in India at least. If you think about it, cervical cancer is number 3, second most common cancer in women. And if you go back and look at the cancer statistics in the developed countries, like United States and in Europe, in 1930s and 1940s, cervical cancer was, top 3, cancer type. So I think with this vaccination and hygiene and other approaches which government and other associations have started, I hope that cervical cancer will become, not top 10 cancer, or people should not worry about cervical cancer in India. These mucins and HER2 actually have been targeted at our, center widely. We have, different clinical studies, clinical trials going on in Nebraska targeting these two proteins in immunotherapeutic protocols. But again, in these solid tumors, most of these immunotherapy success are very limited. In cancer therapy, if you get 15-20% response, basically your treatment will get approval. Think about is how desperate situation is. 15-20% success rate. All these so-called, mega blockbuster drugs, the checkpoint inhibitors, antibodies, they are improving, life from 8 months to 11 months, and not in every patient, So that's what the, what you call it, desperation is in cancer. All in all, at least they are successful. They are showing that they can delay the tumor growth, or at least they can delay the mortality and slow the tumor growth. There are viruses which have been associated with immunodeficiency or different type of cancer, and here are 4 major examples I can give you. Inherited immunodeficiency, there are a lot of individuals. Earlier they used to not survive longer, now, but with all the current treatments we have, these individuals are getting adult and surviving longer, and majority of them are developing lymphomas or hematological malignancies. And in most of these cases, the culprit is Epstein B virus. These are EBV-driven, B-cell lymphomas. AIDS are other immunocompromised individuals which are going through immunosuppressive, therapies. The common cancer types you can find is lymphoma, cervical cancer, skin, liver, or Kaposi sarcoma. And the culprits are all over. Most of these, basically these viruses, infect these individuals and drive, the events which, leads to, different malignancies. Malaria has also been associated with a specific type of lymphoma, which is Burkitt lymphoma, and again it is driven by EBV. And autoimmunity is, again, many of these autoimmune patients are undergoing, immunosuppressive events. If they get EBV infection, the chances of them developing lymphoma is very high. So, these individuals which are immunodeficient, if they get exposed to, a specific type of viruses, they can develop different type of malignancies. Again, here is another example of different, microorganisms which have been associated with different tumor types. Like here, adult T-cell leukemia, which have a very deadly disease. Actually, it is more deadly than pancreatic cancer, and there is no current treatment. There is nothing they can do with these patients. It's associated with HTLV, Burkitt lymphoma with EBV, cervical cancer with HPV, liver cancer with Hep B and C, nasopharyngeal cancer, EBV infection, skin cancer with papillomavirus, and stomach cancer with H. pylori. And in these cases, actually, this is a very important experience, is that, if you can prevent these infection, basically you can prevent, these type of cancer, and which have been very successful. And it is happening as we speak, that, if you can target and block or, do the prevention of infection of these agents, you can prevent many different type of cancer, and that can be exploited very wisely. EBV has been listed many times here, and actually this is one of the very interesting virus which you can use to study your B cell biology. It's very easy to transform your normal B cells. If you infect them with EBV, you can generate lots of different clones. So this is a very interesting a tool if you want to study the B cell biology. One of the cancer which is we have not listed, and we have not even talked about it. Although here you see the skin cancer with HPV. Actually, this is one of the time infection happens, the HPV infection, and most often you can see it's all related with the skin infection. They develop this wart, and this skin cancer actually is #1 cancer worldwide. You know that? Skin cancer is #1 cancer in the world. It is not even listed in any cancer statistics because there are so many cases. In the United States itself, there are 3 million cases of skin cancer every year. Altogether, if you look at incidence of all different cancer types, it comes to 480,000 or something. Okay, skin cancer, 3 million cases. Even in India, I'm sure its incidence is high, but there is no registry. But in developing countries, skin cancer is #1 cancer. But it's curable, 99% cure rate, so it doesn't even make the list. Anyway, that's a side note, that's not immunology. Here is another list of different, major type of tumor antigens, and these are, again, different category. Heat shock protein can also serve as a tumor antigen, cancer testis antigen, which have been exploited. Differentiation antigens with a broader expression. Most of them are mucins and HER2 or other oncogene associated proteins. Common tumor specific antigens like P53, BCR-ABL or papillomavirus, mutated RAS, which have been targeted in pancreatic cancer, unique tumor specific antigens, which are listed here. And I don't know all details about all of these antigens, but all I can tell you is that many of these antigens have been identified in a specific tumor type, and different preclinical studies have suggested that can be developed into different vaccines, tumor vaccine strategies to target those diseases. And here are some of the examples where there are some phase 3, and actually clinical approval has also been given in some certain cases. So all in all, the tumor antigens or tumor associated antigens can be targeted, can be defined can be associated with a specific type of cancer, can induce a specific type of immune response, and can be exploited to develop a specific immunotherapeutic response. So again, we are going through a few of the more examples of different tumor antigens, which we already talked about, which are encoded by tumor suppressor genes. oncogene, different viral proteins, which are depending on the tumor type. These viral proteins are expressed by the, can be presented to your T cells. Oncofetal protein, and the most common one is the CEA, which is widely expressed by majority of the GI tumors, and it has been also used as a biomarker. You can find antibodies against these also, antigens which are present in those individuals which have these tumors. Alpha fetal protein is another one which are predominantly associated with hepatocellular carcinoma and germ cell tumor. This cancer is very common in China and other Southeast Asian countries. Tissue-specific differentiation antigens are, most of them are like, one of them, the most common is GD3. It's a glycolipid identified in melanoma. Blood group antigens have been also identified in some human carcinomas. And more important, there is nothing which have been identified against the metastatic cells, and these are the cells which basically kill the individuals, not the primary tumor. And most of these antigens which I show you here, these are very general, proteases, there is overexpression of different proteases, collagenases or metalloproteinases, which have been associated and linked with metastatic cells. But in reality, there are no specific antigen associated antigens which are associated with metastatic cells. And keep in mind that in metastasis is what kills the patient, not the primary tumor. So I think there is a wide variety of different tumor antigens we talked about, and those tumor antigens can be and have been exploited in immunotherapy. Many of the approaches which are utilizing these tumor antigens are developing vaccines, which can be used both therapeutic and prophylactic. So most of the cancer vaccines are therapeutic in nature, but many of these are now getting used as prophylactic. And the best example of your prophylactic vaccine is your HPV vaccine, which is called as prophylactic vaccine. It is given to the young girls, and to prevent cancer. Other cancer vaccines basically are given as a therapeutic vaccine where the individuals have cancer, and these vaccines have been given to reinvigorate or re-stimulate their immune system. So we will look at some of the popular mechanisms how the anti-tumor response happen. NK cells are major cell type, of the immune system, innate immune system, predominant, component in anti-tumor response regimen, in human or in most of the animal models. These NK cells basically are defined depending on the species you are looking into. In human, they are all CD56 positive, and most of them are producing these immune stimulating cytokines like IL-2, 12, and interferon gamma. They lyse these tumor cells, through the mechanisms which we talked earlier, perforin granzymes or phosphorus ligand, and most of their activity, they are recognizing these tumor cells in non-MHC, defined manner. So these tumor cells don't have to have specific antigens. Actually, if they have antigen, they don't get recognized by, the NK cells. Lymphokine activated killer cells are another group of killer cells which have been generated ex vivo, and this was, in 1970s and 80s, this was a hot item, when the tumor immunology was just getting their foot in the clinic. So these cells were basically, isolating the CD3 positive T cells and activating them with high dose of interleukin-2 and calling them lymphokine activated killer cells. And they were successful in treating melanoma. Actually, they had treated few melanoma cases, and the success of those treatments basically were often associated with non-specific responses. So those individuals who develop these white spots, where they were also targeting melanin expressing cells, those patients did better. So they had response against self-antigen, also other antigen. But they have been targeted, and now they have been reinvigorated, with the better understanding of different cytokines. So LAC cells, these lymphokine activator cells, have come out of the shelf, from 1980s, and now have been utilized again. Different trials are going on, using rather than IL-2 alone, they are also combining it with IL-15 and IL-18, to stimulate other innate effector cells to make this more effective. So LAC cells are not dead. These are, still in different clinical, preclinical and clinical stages. T lymphocytes, you all know that cytotoxic T lymphocytes are important player in providing anti-tumor response. Predominantly it's CD8 positive T cells. We all talked about and discussed. Very effective against virally transformed tumors and also actually carcinogen chemical induced tumors. And they can be isolated as a tumor infiltrating lymphocytes. Most of their anti-tumor responses are associated when the tumor cells are presenting the antigen in context or in association with MSC class 1 molecule. So that's a prerequisite for these tumor cells to get recognized. They have to have antigens expressed in the context that can be recognized by the cytotoxic T cells. And also they have to have this co-stimulatory molecule or second signal in order to get the effective response. So cytotoxic T lymphocytes are another major player in that. B lymphocytes we cannot ignore. We think that B lymphocytes are not playing a major role in immunology, but that's not true. Many tumor types, can be controlled by the products, antibodies which have been produced by the B lymphocytes. Many of these B lymphocytes actually, that they need T cell help, in order to do their response or activity. The antibodies which have been identified, which are related with the tumor biology, many of them has been used as a tool for radioimmunotherapy, or even used as a diagnostic. What you are looking into antibody as a therapeutics in cancer, the market today is close to $50 billion. Okay, 50 billion, not million, billions of dollars. So it's a huge pharmaceutical market. It started with your simple hematological malignancies which express specific markers. CD19 like CD20 and B cells can be targeted by antibody therapies. Radioimmunotherapies have been very commonly used. Our immunotoxins which are attached to a specific antibody can be targeted to the tumor cells. That has been exploited. In general, if you look at just in terms of immune surveillance, at least in preclinical model, you will find that if you eliminate, of course, we all talked about CD8 positive T cells and how important they are in preventing cancer. Okay, but there are experiments you can design to show the significance of B cell in itself, where you can remove the CD8 positive T cells and then transplant the tumors or implant the tumors. And you will find that still those animals which lack CD8 positive T cells have also will allow not allow these tumors to grow. And in those cases, the responses as because of the B cell, so CD4 B cells can in itself in certain tumor types can take care of the tumors in absence of CD8 positive T cells. So experimentally we have shown that B cells are not just a bystander. But of course, definitely the products from B cells, specifically the antibodies which have been, immensely utilized in the immunotherapies. Macrophages, this is where my journey started in cancer. Basically I said that, in 1987 I started my PhD work, and we thought that we can cure cancer by just activating macrophages. And that was actually not a bad idea, because if in vitro we can do it, in vivo there were also evidence to support that activated macrophages can inhibit tumor growth. And one good thing about these cells were, like NK cells, that these cells, they don't need a specific antigen. They can, target any tumor cells, and they recognize tumor cells not through antigen or not through NK.1 or other NK target. They recognize antigens through a whole different biochemical phenomenon. Those phosphatidylserine or phosphat, it all depends on what is on the transformed cells. They can get any transformed cells can be easily identified by the tumor macrophages, and those macrophages basically they can engulf those tumor cells and eliminate them. So there was a hype at that time, and we Actually, not, so far actually, if you look at its utilization in immunotherapy, it has not been utilized. Now, the approach is very different. Now they want to block the macrophage infiltration in the tumor rather than bringing them in the tumor, because these macrophages, most of the tumor-associated macrophages, are enhancing tumor growth rather than inhibiting tumor growth. The only cancer where the activated macrophages has been used as a therapeutic is an osteosarcoma. Osteosarcoma is the only cancer where the macrophage activating agent has been utilized to treat osteosarcoma. There is no other treatment. This is muryl dipeptide, this derivative of this muryl dipeptide, which have been used in osteosarcoma, activates macrophages to remove the tumor, so which has been used. But it got a break for me. At least I went over there, learned a lot, and continued and learned, tumor biology, learned about immunity, and is still trying to learn more and more about it. But I have not given up on macrophages. Still, we continue to work on it because, these are universal cells. If you look at, they are everywhere, and in tumors, you can find all tumors have it. So I think we will discuss more in tomorrow or day after when it will come to macrophages and dendritic cells. Most of these cytotoxic events which happens, as I said, I kept talking about these porphyrins and granzymes, and the basic phenomenon how they happens, this pore formation happens, is very defined cellular process. Where the activation, once the engagement happens, once the antigen is recognized, there's a trigger which causes the release of these granules, and these granules complex together to form these pores. And once these pores are formed, basically the one thing happen is that once this pore, the barriers is broken, and most often these cells go under necrosis. These are extrinsic pathway, and it basically induces necrosis in most of these tumors. There is events where the death ligands have also been directly involved, and this can be your NK cells or your CD8 positive cytotoxic T cells, which express these FOS ligand. So FOS is expressed on the tumor targets, or TNF receptor is expressed on your targets. If these two, cytokines or these ligands are expressed, they can get engaged and can induce. This is apoptosis, which will be more like a programmed cell death. Actually, FOS ligand is also involved in T cell homeostasis also. But tumor cells, many of them, they do express FOS, and if they do express FOS, they can be easily targeted by FOS ligand, which are expressed by NK cells or cytotoxic T cells. And this process is often mediated by apoptosis. That porphyrin granzyme pathway is most often associated with necrosis. Many of the cell surface molecules on T cells, NK cells, or macrophages have been exploited for immunotherapy. The legend is very long here, and I left it knowingly. I think some of these slides, I think if I will let you guys upload it, if I will just leave it. If you guys want to, you can print it out. But all in all, I'm showing you here is that if you look at the T cell in itself, many of the molecules which are expressed on the T cells has been exploited extensively for immunotherapy. You all know about co-stimulatory molecules, which have been targeted, and antibodies have been developed against them and to block or enhance these responses. Checkpoint inhibitors, PD1 or CTLA4 and PD-L1 has been also exploited. T cell receptor, now they are trying to create these, chimeras where they can exploit the specific T cell receptor if they know the tumor antigen, and they can drive these T cells to specific tumor types, have also been exploited. In terms of NK cell, activating receptors have been utilized to develop immunotherapeutic protocols. Fc receptor, which is CD16, it's not just restricted to NK cells, but in NK cells it is a predominant mechanism to induce antibody-dependent cellular cytotoxicity. And one of the question I had earlier to you, that how NK cells recognize these tumor cells which don't express antigen, these killer immunoglobulin receptors, which are over there on NK cells, are the important mechanism in that. In terms of macrophages, they are not dead in tumor immunotherapy. They are still very much functional because most of these tumor cells, what they do is that they start expressing some of the molecules which allow or which basically tells the tumor cells that like here checkpoint inhibitors, they are trying to block the T cell responses. In this case, basically they tell tumor cells not to eat me. The basic signal is that, because macrophages are very good at eating anything and everything which doesn't look familiar to them. Okay, so this CD47 is a very interesting molecule. We try to block CD47, which basically will block this whole don't eat me pathway, and can activate this whole macrophage dependent tumor cell killing. Or macrophages can be activated through the Fc receptor complexes, or can be directly activated through different macrophage activating factors. Antibodies-based immunotherapies have been also very widely studied. It can be directly through the antibody-dependent cellular cytotoxicity. There are different bispecific antibodies which have been also developed, which can target both the tumor cells and can bring your T cells and NK cells to the site of tumors. Many of these antibodies which have been used to target the growth factor receptor, and best example is your Herceptin, which blocks the HER2. Or these antibodies can be conjugated with radioisotopes or chemotherapies to target the specific receptor within the tumor. So antibodies has also been passively utilized to develop different immunotherapeutic protocol. So in general, the simplified version of the summary I can give you here is that in most often normal cells are normal. They don't get recognized with the tumor cells. But if anything happens, some insult which causes the tumor cells to get transformed, they get recognized by your cytotoxic T cells or natural killer cells or macrophages. Immune cells, they are constantly monitoring our body, surveying our whole system, trying to find out if there is any transform event or transforming cells and eliminate them. Tumors develop when this surveillance fail, breakdowns, or when it is overwhelmed. And of course, there are a lot of ways we can develop targeted immunotherapies, and hopefully, we can manage malignant disease better. But again, there are a lot of hope. Some hype is there also, but all in all, I can tell you is that tumor therapy is going to be the future. Okay, so this was actually a very general overview of the tumor immunity which we covered. Okay, next lectures which we will have will be more specific. We will cover one area where we will basically talk about inflammation and how it impacts tumor immune response. Other one we will have more focused on antigen presenting cells, macrophages, plasticity, and other checkpoint inhibitors, different immunotherapy. So we are going to just divide these different lectures into different classes, and hopefully we will end with some future perspective at the final day. Okay, so that's our general strategy. Make sense? This is our campus where I am actually. So this is a very nice picture. This is the latest one. This is the cancer center which was built 2 years ago. This is the outpatient clinic, and there are 2 towers here. Basically, these are the research buildings, 10-story tall. So these 3 buildings together here, there are over 500 labs over there. So all the basic research, is focused on here. In cancer center, they have like 2 wings. One here is the basic research, and on this side is the patient care. And so there is they have put it together so that at least there will be a dialogue between clinic and basic research more effectively. So physical barrier they have tried to remove by doing that. And actually, my lab and office is here, on this floor, and 8th floor actually. So I'm way high over there, and so I think I have a better view of the whole campus. And this is main entrance for the hospital, and that's all. I will be happy to answer any question, and we have time for interaction. I'm available. Okay, I'm not leaving anywhere. So anytime if you have any question, feel free to come and talk to me. Okay. Thank you for your attention.